Okay, welcome back to the last of the workshop sessions of the day. My name is Geoffrey Fudenberg. I'm a professor at USC in the Department of Quantitative and Computational Biology. And this tutorial is developed with Paulina and Yao, students in the group. They're sitting in front. So the format will be a little different. I'll instead, since we're a fairly large group, I'll instead start with some slides, and then we will break into groups, and I will tell you how to do that when the time comes. In your groups, you will work through the tutorial together and collate questions, and then we will go through as many as we can in the time that we have. And then, of course, if you have additional questions after our time is up, don't hesitate to discuss. We're here for the week. So with that, thanks to the organizers and all of you for attending. Hopefully we can make this high information content and fun. Here we go. So let's say our goal is to have some predictor of what a contact map might look like from DNA sequence. So I should say that the background assumed for this talk is just that you were here for the other tutorials. So some of you may be experts, others of you may be just getting on board. So that's obviously something we're trying to bring everyone together. But we all saw Sasha's tutorial, so we all know what is on the right, and we all maybe are curious about how the DNA sequence that we have can instantiate such a genome folding pattern. This is about a few megabase region of a human genome. They all kind of look like that if you scroll around. And the challenge of this problem is that there's a lot of DNA, right, which can be illustrated many different ways. Here, a few different motifs for CDCF are highlighted in this 2048 base pair window, which is highlighted again on this contact map. It's one bin on the contact map. But the real challenge, or the real kind of point where it starts even getting interesting, is that we need to not just predict what's happening in just a bin of this contact map, we need to predict what's happening over about 500 bins of this contact map, about a megabase, where these features start to emerge. That's a lot of DNA, okay? And there are many, many things that look kind of like CTCF motifs in this million of base pair windows. So how would you pick up the ones that actually matter and then transmit that information faithfully over long genomic distances and come up with something that can actually make predictions? So I'll show you one way which you can do that. This is a growing field. There's many emerging new ways to do this. So don't think that what I'm telling you today is the only way to do it, but think this is one of many interesting possible ways to go about this problem. We're actually, what we're doing is remarkably similar to an art project here performed by the artist Matt Bierner, who you can look up the video on YouTube. But he decided to print out a chromosome onto punch tape, and he actually had to stop his experiment after chromosome 2. But what you'll see is that how we're dealing with DNA is actually very similar to how this artist is dealing with DNA. We're going to end up encoding DNA as a set of ones and zeros that is eaten in by a computer, and then we are going to try to make predictions with those numbers. All right, everyone on board? Good, okay. So, and the way that we're doing this is with neural networks. So they're very popular, but the general idea is very simple, okay? The general idea is you have some input, some set of numbers, and we call this x. We're going to learn some set of transformations from the data, f of x, to predict some output, right? We're just going to approximate some output. So for computer vision, a very classic problem in this field, you might have the problem of classifying a handwritten digit, right? So you are going to take in an image as a set of pixels with values for a grayscale image like this 2. Each pixel is between 0 and 1, and the output is a probability maybe that a number is between 0 and 9. Of course, the internet is made out of cats and dogs still. So you might be able to also design, you might want to design a network with all that training data to take in an image and tell you if it's a cat or a dog. Of course, this one is I don't know, to me pretty challenging, but you can be the judge of that, or let the neural network judge. So what I think is very appealing about these neural network approaches in general is very flexible. So you can use a similar strategy in many different applications. So here, and I think this is a nice illustration from computer vision again. Here you have image classification. You want to classify what's in it. There's a person with sheep and a dog. Maybe you want to, a related problem would be object localization. Where are the objects in the image? Where are the sheep? Maybe even further, you want to go a little bit further and you say, oh, what are the objects where in the image, right? So this is semantic segmentation. You can do all of these related but distinct tasks with the same sort of approach. And what I find motivating about this and seeing what's been achieved in computer vision is that I think in genomics, we've only kind of scratched the surface of what can be achieved with neural networks. And maybe we can step back and think by analogy and come out with some new ideas from this workshop of how to apply neural networks in genomics. There's, of course, very fun examples of neural network applications like style transfer. You have an image, you have another image, you try to transfer the style from image to the other. There's also emerging uses of these neural networks that are maybe more useful. For example, this is from a paper integrating spatial gene expression and breast tumor morphology via deep learning. And there's a lot of promise in medical image analysis, in predicting outcomes to chemical reactions. And of course, you've all seen probably AlphaFold and protein folding as these sorts of predictions are now routine to make with neural networks. This is actually from a paper that instead of predicting a 3D structure of protein is predicting a contact map. So it's actually, there are kind of related ideas in the protein folding literature to also draw on for these neural network approaches in genomics. There's some interesting kind of sharing of ideas. Okay, so what's the incredible idea? So to me, this is really an incredible idea that if we have enough pictures of cats or not cats, we can learn how to detect them just from the data, right? So we can train a model to learn the best set of transformations of the input data for the task. And the very kind of rough gist of this is the network makes a current prediction. You compare it with a ground truth. You use that comparison to update the parameters of this complicated set of transformations, and then you provide the network with more input data. And you keep on doing that until some measure of performance is satisfied. And we'll talk a little bit more about that in the second part of this tutorial. Okay. So, of course, it's not quite that simple. There might be some challenges associated with each of these steps of the neural network. Some of them that are always going to come up. So if you're reviewer 3 on a neural network paper, you can kind of have this checklist. So for the input, how did the authors pre-process the data? What are the type and number of transformations learned that they're asking the model to learn from the data, and how can you evaluate the performance of your model, and how can you interpret what this network is doing or not? These are something that always want to make clear. So we're going to try to do that today for our particular example of genome folding. And as we go through this, so we're going to go through these ingredients in this order. We're going to first talk about the architecture of neural networks, activations, layers, loss functions, the data, so that includes preprocessing and data augmentation, a little bit about training, implementation, and valuation of neural networks. So, and I'm going to focus on the architecture first, going from a convolutional neural network for computer vision before moving to genomics, and then to training, etc. Okay. So this is an example, it's a cartoon of a neural network that crushes the handwritten digits task. This is a convolutional neural network. And what it is doing, again, is to take a low resolution picture of a number that's been handwritten and assign a probability that's a given digit. Okay, so you might have seen some complicated pictures of neural networks like this, but I think part of the beauty of these approaches is that the fundamental ingredients are very simple, right? And you often mix together very simple ingredients to get a kind of this very rich behavior in a final model. So building up to the full architecture from individual units, the simplest thing that you can do is would be you have a set of inputs, you have a set of transformations, which are just a set of weights multiplied by your inputs, giving you an output, right? So this would be just a summation, a dot product, a linear transformation, whatever you want to call it. Or you can represent this as a matrix multiplication. However, neural networks typically use nonlinear transformations, okay? And the sort of the simplest, arguably simplest possible nonlinearity would be a step function, right? If you're below a certain value, you're nothing. If you're above a certain value, you're something. But this sort of function is hard to optimize the computer, right? So what works a little bit better? This is the story of neural networks in a nutshell. What works a little better? So a common nonlinearity that you'll encounter is the sigmoid. This is what's used in logistic regression, for example. The sigmoid function, right? It's smooth. It starts at 0, and then it saturates at 1. It has some nice kind of linear region connecting these two saturation regimes. However, we don't use the sigmoid for neural networks typically because the output is not centered, and neurons, their output is saturating both at the low end and at the high end. So it's hard to take gradients through these. It's hard to update parameters of your model if you use this sort of activation function. So in practice, the nonlinearity that is used in many neural networks and what we use, though of course people like to debate about these things and argue about which is the best thing to do. What we use is this ReLU activation, which is maybe the second simplest nonlinearity. It is 0 below 0, and then it is just the input above 0. Okay, so if it's the advantages, it's inexpensive to calculate. Of course, it's not without drawbacks either. Okay, so that's nonlinearities. We're done with that. And we're moving into layers. So the simplest layer would be a dense or fully connected layer. And we saw one when we were describing activations, right? You'd have a set of inputs, you multiply them by their weights, and sum them together. You could also add some offset term. That's a common thing to do. So my output after this first layer is my set of weights multiplied by my inputs from the first layer. And we often cartoon this as a dense set of arrows between this input and this first layer, meaning that every unit or node in this first layer is receiving an input from every single, from each of the 3 input numbers. Okay? Questions? No comments allowed yet. We can make neural networks more expressive and learning more complicated ways of processing inputs, either by adding more nodes to a given layer or stacking these layers on top of each other before computing the output. So this would be a schema of 2 dense layers, one after the other, where we're learning a set of weights, that a 3 by 4 set of weights to connect from the first layer to the first hidden layer. Then we're learning another set of weights, now 4 by 4, to go from this layer to this next layer. And then we're learning another set of weights, 4 by 1, to make a single prediction for, say, 3. So here we'd have 3 input numbers in our x, and then we'd have 1 output for our f of x that would be compared to what we're trying to predict. And just one thing to note is I should have said this before, but we're considering feed-forward neural networks here. So you notice that there are connections between neurons across layers, but not within a layer and not to previous layers, right? So we're all this feed-forward property, asserting this feed-forward property on the types of models you design, makes it tractable to quickly update their parameters and find good solutions. Okay, so instead of allowing like every position in the previous layer to influence the next layer, a common thing that you want to do is you want to restrict this to a subset of positions. And a very natural way to do this is with convolution. So in computer vision, from images, uses 2D convolutions. Though we'll see kind of mostly 1D. I'm trying to pause this animation. Can't do that. Though we'll mostly use 1D convolutions in genomics. What is this illustrating? It's illustrating that each pixel, each in the second layer here in green, levitating above the first layer, is influenced by a 3x3 region of the previous layer. Okay, so instead of a dense layer where you're kind of taking information across every possible node in the previous layer, you're saying, okay, I'm only going to take information from some local neighborhood of pixels in my previous layer. So that's one of the most common things used in computer vision. It's also one of the most common things used in genomics because we are inspired by work in other areas of science, and have not come up with new layers that to this day, I don't think there's been a layer developed in genomics that has kind of gone back and had a big impact in computer vision, but you know, the future is next, so who knows. Okay, the next layer to understand for these sorts of networks is a max pooling layer. And this is also very simple. It is saying that for each position in the next layer, I just take the maximum value of some local neighborhood. This would be a width 2 max pooling, 2x2 max pooling. So I take sets of 2x2 pixels and I pass the maximum value on as my value to the next layer. So why would you want to do that? Well, it's a natural way for a network to ask whether a feature is found kind of anywhere in a small region of an image rather than like exactly at a certain position. And another part of this intuition is once a feature's been found, its exact location isn't as important as its kind of rough location relative to other features. And one benefit of this pooling is that there are many fewer features after pooling. Right here we've gone bound by a factor of 4, and this can help reduce the number of parameters needed in later layers of a neural network. So very common strategy in computer vision, very common strategy in genomics. Okay, why might all this work? I wanted to give one statement, which is that feed-forward neural networks can be thought of as universal function approximators, which means for a suitably smooth function, if you have enough hidden units, you can approximate it to any level of accuracy. If you go to one dimension, you can kind of get an idea is that you could use different nonlinear units to make bump functions, and if you have enough bump functions, you can build up a curve. Okay, so that's why it might all work, and you can read chapter 4 for more information if you're curious. Okay, so we understand this, hopefully. Hopefully everyone now can understand this schematic of a neural network that can kind of defeat the MNIST challenge, where there is convolution, followed by pooling, followed by convolution, followed by pooling, followed by some fully connected neural network combining all of these what we call features, and another fully connected layer to give us 9 outputs, okay? Which can be compared with our 9 numbers that any given input could be. So I don't know, I like to think, how many people used Legos growing up? I don't know. Some fraction. So I like to think of these networks in some way as similar to Legos, right? You have these different building blocks, so you can put them together and with some flexibility to achieve the goal of your design goal. Okay, any questions before proceeding about computer vision kind of whirlwind tour? Okay. We'll have more time for questions later in small groups, so then we'll also be, well, that might be easier way to ask it than in a larger group. Okay, so, okay, how could you do this practically? In the computer, we need some practical way to put layers together and compute loss functions, update parameters. This is what this sort of network would look like in Keras. This is a very popular framework for building neural networks. We make use of a bunch of Keras layers for the network that I'll be talking about later, this Akita network. And you can see just defines, they have convolution layers, they have max pooling layers, they have some dense layers, put them together and make some predictions. Of course, you need some data. You can download the MNIST data if you're curious in this sort of thing. The Keras tutorials are really good. I recommend going through them on your own, understanding them. You can learn a lot from how they design the tutorials. You can learn a lot. I learned a lot from how they actually design their code base. It's a really beautiful piece of work. Once you have your data, your code, you train your model, the next important thing to do is evaluate it. And we often report some quantitative performance of the model, right? Like how much does my prediction correlate with the real data. It's also very important to look at the qualitative predictions of your model. Does it, when my model is getting something wrong or somewhat wrong, how well is it doing? So here is an example of ImageNet, which was a very influential model in the literature. It uses large sets of labeled images. Has 8 layers with weights. The first 5 are convolutional, the last 3 are fully connected. And then it has a 1,000-way softmax, which produces a distribution of over 1,000 different class labels. So you can see, so this is what would make me happy as a researcher, is that my prediction for leopard, not only is it doing pretty well, my model's saying that leopard is probably what this thing is. The off-target predictions are like jaguar and cheetah. So I mean, that's pretty good. This motor scooter, okay, it's a go-kart or moped. This might, sure, it could be a starfish. I don't know. I'm pretty. That's the sort of thing where I'm pretty happy. It's off the center of the image. Maybe it's more challenging to identify. So I would go through the predictions of my neural network, take ones that are really good, really poor, and see like is what the model's doing making any sense or not. So this is the importance of qualitative evaluation. Another thing that's fun to do with these neural network models, hot area of research, is trying to interpret what these models have learned. So this is just a plot, I think also for ImageNet, of the filters, these convolutions that are being learned at the first layer. And what you can see here is that some of them look like edge detectors. So you can see these little black-white zebra stripes. And then some of them look like color detectors and color contrast detectors. We have like 2 different colors next to each other, color gradient from pink to green, color gradient from orange to blue. And these are the things that are being run over the image at the first layer. So things that we would have designed by hand maybe 15, 20 years ago as, oh, like an image should be made out of edges and colors, are things that you can, if you have enough data, just learn from the data itself. And I think that's a really exciting concept, and I think that's something that we're also just starting to scratch the surface of in genomics. Okay. Which brings us to a brief history of neural networks in genomics, which I call F of DNA neural networks, and a chance to pause. So if you are planning to use the collab for the tutorial portion of this workshop, please open the collab and run the first cell to install the dependencies, because it takes about 7 minutes. Sorry, then the first cell has to be finished before the second. Yes. The first cell has to be, so we have to run the first cell, has to be finished before running the second cell, which will take another few minutes. But the first cell is the really slow and important one to do first, because it's setting up a conda environment with a few different dependencies for running any of the software. And if it doesn't finish, then the next cell will just execute and say, oh, you didn't install the packages. So just run the first cell and wait for a second, maybe 5 minutes, and then run the next. I won't be silent for 5 minutes though, don't worry. Okay. So hopefully everyone's conda is happily chugging away, installing dependencies. If not, we'll be working in groups, so hopefully someone nearby you, spatially, that worked with them, and you can work with a friend. Okay, in genomics, the first, to my knowledge, the first main class of neural networks were seek-to-peak, is what I like to call them. So they're taking in DNA sequences, or at least FFDNA neural networks. We're taking in DNA sequences, one-hot encoding them, right, like as a punch card, and then predicting some output, usually from a technology like chip sequencing, which is saying, like, is this piece of DNA bound by a particular protein or not? And the transformations were just convolutions and pooling and dense layers, just as we talked about for the MNIST challenge. So you can just as you can do very well with the MNIST, you can do very well with the these sort of seek-to-peak challenges with very simple set of operations. The next class of models in the literature were sequence to profile models. So the sequences got a bit bigger, 100,000. The transformations got a little bit fancier, more on that in a second. And the loss function got a little different. But they also work quite well, as you can see for this example of a histone mark. So this is, again, this is 100 kb or so of a held out portion of the data set, the test set. The experimental data is in the top row and the predictions below, and you're getting not only the position of these features, but also the relative intensities. So that was very exciting to me. So what are these couple of tricks that they used? One is a dilated convolution layer, same idea as the convolution, except instead of taking just adjacent pixels and passing those to the next layer, you're taking pixels spread out by some dilation factor. Here the dilation factor is 2. That's actually also the same dilation factor that we often use in genomics. And combining with that is what's called a residual connection. So you not only do you do this dilated convolution and pass the output of that forward, you also feed forward the previous layer, and apparently this helps training deeper neural networks. It's a trick that worked well, I think, first in natural language processing. It also worked very well in genomics, in 1D genomics. And then one thing that I wanted to highlight is that this interpretation aspect. So this is a plot of, for one of these sorts of networks, of influence versus information content of the first set of filters from this network. And basically what they did is they just took the filters that were learned by the neural network and then compared them to the database of transcription factor binding preferences. And what you can see is that it learns things like FOSS, like it gets this TGA spaced in TCA. That's useful for the model for predicting a large compendium of ChIP-seq profiles. Or you get this GTAAA. It's useful for predicting some of these large set of profiles. So this is one way that people have tried to interpret genomic neural networks. Another thing people have used in genomics is use them as tools for interpretation of other data sets. Here is an example where there's thought to be some particular nucleotide that's important for vitiligo, and the researchers made every possible mutation, computed the influence of every single base pair around that position, saw that there's this little cluster of what are called salient nucleotides, ones that are important for the model's prediction, and saw that that allowed them to interpret the genetic data and say this is probably a binding site that was disrupted for a particular transcription factor. So that's a sort of way that we might hope to use neural networks in a downstream application. This is how people are already using them with kind of these 1D genomic neural networks. Maybe there's ways to use genome folding data as well for this sort of challenge. Okay, which brings me to this sequence to map challenge, right? Where we're taking in DNA sequences, We again one-hot encoded them. We want to learn a set of transformations that can predict sort of a patch of a high-C contact map. And you'll notice here, instead of the fall color map that Sasha was showing us, this is a blue to white to red color map because we are predicting the contact frequency pattern after removing this effective genomic distance. So, and they were actually predicting the log of that. That turned out to work well in practice. And there might be ways to predict the observed data, but it worked well in practice to predict this pattern after genomic distance dependence has been removed. And just as the sequence to profile models involve some additional layers to make that work well, we also had to add some layers to make this genome folding problem tractable. And the most important thing is we needed some way to convert information in 1D to 2D, right? Because instead of predicting a profile from a 1D sequence, or predicting a map from a 1D sequence. So there's many different ways that you could imagine doing that. You can imagine taking an outer product, or a geometric mean, or concatenating information from position I with position J. We tried all of them, and it turned out that just averaging the set of information from position I with the set of information from position J worked very well. So that's what we did. Okay, but you need some way to convert from profiles to maps. This is the schema of the architecture that we ended up with. You have DNA sequences, that's our input. Transformations include convolutions with ReLUs and pooling, dilated residual convolutions, and converting to 2D, and then more dilated convolutions. Same as before. Any quick questions before I continue? Okay. So what does this network let us do? What do these networks let us do? These are examples of predictions from the model on a test sequence. So DNA sequence that hasn't seen held out regions from the test set. In the top row is the real data. On the bottom row is the prediction. You can see it's able to learn some sort of strong boundary where there's a strong boundary, not much going on when there's not much going on, more going on when there's more going on. Again, this is about a megabase by a megabase. And we quantified our performance. So in addition to the qualitative performance, we also did quantify the performance, and we used two metrics because we wanted to be able to both correctly predict very flat maps as well as very feature-rich maps. So you can see that you can have a good prediction in terms of the MSE, even if there's not much going on. So sometimes it's useful not only to evaluate your model qualitatively, but it's also kind of useful to evaluate performance with multiple methods quantitatively. So that's another, if you're reviewer 4 now, maybe you want to ask for or think about, well, why did they show this measure of performance instead of some other one? Okay. So we talked about, we did architecture, we did evaluation, interpretation. So what can we do with a model that takes in DNA as an input? We know CTCF is important, so we can mutate CTCF motifs and make new predictions. So that's what we did. You can see here experimental data, predictions for those regions, decent predictions, and then if you get rid of all the CTCF motifs in their window, you can see you get these relatively flat maps. So that's one way that you can additionally evaluate your model by challenges beyond the test set. Is it going to reproduce qualitative features of the data that I care about? In this case, genome folding. Okay. Another thing that you might do is looking at mutagenesis around CTCF sites. Which positions around CTCF sites are most important? You can go nucleotide by nucleotide. You can compare a resulting profile of influence with a measure of evolutionary conservation. So this is a Philo-P score. So that was a way that we could say, well, maybe the influences at the single nucleotide level that this model is picking up are interesting for people to think about. Last sort of now. in addition to understanding current experiments, you can also use models to sort of generate hypotheses, right? Like I like to think about these neural network models as hypothesis generators. So this, we call them experiments in our group that we're a dry lab. We call them computational experiments, is involve throwing mutations just randomly across the genome, setting a threshold and saying mutations above this are impactful, and then looking at where they fell. Many of them fell into CTCF as expected. A bunch of them fell nearby CTCFs. We're kind of trying to understand that now. You can go check out a poster and talk from Paulina about that. And then many of them fell into other categories that are not yet annotated. So there's a lot to learn. At least 20% of the impactful positions, according to this model, are not clearly annotated for predicting genome folding at the megabase scale, which we already know a lot about. So I think that's pretty exciting. We're maybe 40% of this puzzle, according to this model. Okay, so yeah, I wanted to say there's also there's many emerging methods for doing this: DeepC, Orca, Chromofold, Epiphany, and many more. A lot of exciting ideas, alternate decisions on what layers to use, on what loss functions to use, on the shapes of what pieces of the contact map to try to predict. And yeah, you can go check out posters from Yao and Paulina later this workshop on developing and interpreting 3D genome models. What I wanted to do now is this is just sort of the warm-up to the tutorial part 1. So hopefully your collab notebooks are kind of mostly working by now. How is everyone's 50%? What's our success rate? Okay, yeah, like 40%. That's good. So again, we'll talk about the architecture first in the first part of the tutorial, and then we will go into training. We'll come back for some more slides, and then we'll go to training in the next part. So just to quickly preview, this architecture I like to think about it in terms of a trunk and a head. So this trunk is very similar to these seek-to-profile models, very similar to the computer vision models, where we're taking in a DNA sequence, we're one-hot encoding it as ones and zeros, we're passing it through convolutions, nonlinearities, pooling, and then we're converting to the land of maps from the land of profiles. And that brings us to the tutorial part 1. So what I want to do is I would like every other row to flip around, meet their neighbors behind them, and then we'll split the room in half and we'll have 6 groups. So the people in the very first row, we will disperse you throughout the audience. You will find new groups. But this second row here, if you guys can turn around and look behind you, and look what half the room you're on. The people on that half of the room will be on a group there. The people over here will be in a group on this side. Same with the fourth row, we'll flip around. And same with the row after that. And I will see if everyone has enough people in their group and reassign people to groups if that's not the case. In your groups, you have one assignment, which is to make it through the motif insertion portion of the notebook. And I want you to write down questions that come up with your group, and we will try to go through as many of those questions that come up as time allows. Okay. And Paulina, Yao, and myself will circulate. So you can also, if you have a quick question, as well as Ed and Sasha and maybe others, if you have issues with Conda, like wave a Conda hand. If you have other questions, write them down and share them with us. Oh, and introduce yourselves, right? You're here to meet people as well. So yeah, say hi to your neighbors. Paging the workshop. Well, let's get started with the second portion of the last session of the day. I'll give you a second to get back to your seats. While doing that, let's go over a few questions that people had. Okay. What exactly was permuted and how does the permutation work? So for predicting a genomic sequence, what we did for permuting sort of obliterating the influence of CTCF sites is we just scrambled the nucleotides randomly. There's many ways you could imagine doing this. One challenge is, again, is comparing with experimental data. There's different ways to disrupt genome folding, and maybe you want to model those different ways of disrupting genome folding experimentally, differentially inside the computer as well. We haven't done that yet, but that would be interesting. Like, for example, CRISPR-I, you might CRISPR-I a CTCF site, and that might have a different impact than like deleting it, which might have a different impact than using a base editor on the CTCF site. For now, we just said, okay, what's the simplest way we can just, on average, obliterate a CTCF site? But there's a lot more to be done there to actually make maybe better connections with experimental data. Color bar ranges were in general, I think, like -1 to 1 on a log scale. This is sort of a natural scale for these high C maps after removing the influence of genomic distance. What is a convolutional tower? That is just a set of convolutional layers. It is a shorthand. So for when you're designing a neural network and you want to say, one thing that we didn't talk about is that when we're training a neural network, sometimes you want to explore different architectural choices. Maybe you want 10 layers or 12 layers. So if you want to be able to easily do that, you need to have your code designed in such a way that you can sweep over, say, the number of layers. So this tower function is a convenience function to enable sweeping over the number of layers. Okay, another question was, why is the output array whatever by 5, and the final high C is 500 by 500? We predicted, we only kept the upper triangular half of the matrices, so that just for space, save half your storage, half your memory footprint. The two, and that brings me to something that was not asked by anyone, there are two memory bottlenecks for these sorts of networks, which is at the very start of the network when you're inputting DNA, and at the very end when you're predicting maps. Because when you're inputting DNA, you have a very long sequence before you've done any pooling. So it's very long. So the size of the object you need to store in memory is large. And that's actually was the main bottleneck on the GPUs at the time that we used at the time, which had like 12 gigabytes of memory. The other bottleneck is if you try to predict larger and larger square matrices, and you have, say, many different filters that you're using to predict that matrix, you also can have a memory bottleneck at the end as well. So it makes sense to kind of just predict the upper triangular half of a symmetric matrix. So that also gets a little bit into another question, which is why use pooling? So yeah, pooling lets you both have some translational invariance, so you don't care exactly where a feature is, but kind of more its relation to other features. But it also lets you have networks with a smaller memory footprint. That's one other kind of benefit of doing some pooling. And ultimately, we have to compare our predictions to data that's only really able to be analyzed at a certain resolution. We'll talk more about training coming up. One thing that came up for a couple of groups was, what are these stochastic shift and reverse complement layers? They are to do data augmentation. I didn't explain them yet, so glad you asked questions about them. Thank you. We also had a question of why is this predicted map so smooth, and why is it so much smoother than the experimental one? That's an interesting question because experimental data, right, we're limited by how deeply we sequence with our ability to get something close to the truth about genome folding. In models, we don't have any stochastic component, so it appears smoother. Okay, then how to quantify the goodness of the prediction. Let's come back to that after training. Can you generate a given sequence from a contact map? We haven't yet. That's a very interesting area in sequence design. A few groups are curious about that. So if you have ideas, get in touch. We'd love to try them out or work with you. And the last one I'll talk about is, what about genome folding in different cell types? So this model that was presented here was jointly trained on the 5 different human cell types. They didn't have tons of different, they're all cell lines. They didn't have tons of different genome folding differences between them. The model didn't learn huge differences between the different cell types, so we weren't able to extract anything interesting from a sequence basis that differed among these predictions. There's a little bit more in a slightly updated model that Paulina will talk about and has kind of more analysis on in a poster, but still minimal. So I'd say that's a kind of an open, interesting question, is how, because we know genome folding does vary at specific loci between different cells. What's the best way to capture that in this sort of sequence model, and what sort of data sets are needed to actually really capture that? And I think those are interesting questions. Anything else related to mostly architecture? Or if not, then we'll move on to more about training. Okay, training. So yeah, this is the same overview picture that we had before. What are we doing? We are taking input data, learning transformations, generating an output, comparing it to some ground truth or labeled data, updating parameters, and feeding our function approximator more data. The first thing that we need to define after the architecture is how our training data is processed. So here, for this network, we took data processed with a distiller pipeline available from Open2C. We enjoy working with, processed into coolers, binned at a particular resolution that seemed appropriate for these different data sets. You'll see a few micro-C data sets, a few high-C data sets. Then we smooth the data because it was still relatively noisy in terms of there's still sampling noise at that, as noticed by folks in the audience and the experimental data. Then we took observed or expected with CoolTools functions, as Sasha discussed, and then so neural networks don't really like NANs. So actually, this is something that the first iteration of models that we tried to train failed on, is we didn't handle missing bins very well. So one solution that we found that worked was if you have a big stretch of missing data, like a centromere, right? We have no DNA, we can't get high-C data for a centromere right now. We truncate the chromosome there, so we don't try to feed that to the neural network. If we just have a few missing bins, we interpolated the missing data. So otherwise, the network just learns where missing bins are. They're very powerful, but also, the people like to say garbage in, garbage out. It's very true. Okay. Yes, we did standard augmentations for genomic data sets. We use what everyone else uses, which are shifts that are much less than the bin size and reverse complementing of the whole input sequence. And then we said, okay, in order to decide how to update your parameters, you need some measurement of the difference between the output of the neural network and the true data. So for this handwritten digit task, right, you learn some transformations that give us numbers between 0 and 1 across a set of from 0 to 9, and that has to be compared with 2. That people usually use a cross entropy for that. What we used for Akita was just the mean squared error loss between the model's prediction for a megabase by megabase patch and the true data for that megabase by megabase patch, because we were predicting something that is in this kind of -2 to 2 domain. Okay. Right. So then this In the JSON that define the architecture, we also define you also can see the parameters used for training. You can see there's a batch size of 2. I told you about memory constraints based upon the size of the GPU you're using. We'll talk a little bit more about the optimizer. This is SGD, stochastic gradient descent, the learning rate and the momentum, parameters of how this optimizer is doing its parameter updates, the loss function, which we just described, mean squared error, just some of the squared differences between your true and true data and experimental data. Clip norm is a truncation on the updates to this SGD. So if your update would be huge, don't do it. And this patience term we'll talk about in a second. Okay, this is how it's implemented at the level of code. We're just using, like I said, we're relying on really an amazing body of computational knowledge and literature baked into these optimizers. This is this TensorFlow Keras optimizers, SGD. That's what we're using. It lets you pass a bunch of parameters to it. We use the same optimizer that's used for driving cars around the street, which is a little scary. Okay, so how do we update parameters? So this is just a schematic. So imagine we have a neural network with a certain set of parameters at a certain point in time. Maybe your first step, you initialize the parameters randomly. You make a prediction with this F of DNA. We have a predicted Y, predicted contact map. We compare that with the true labeled contact map for that piece of DNA, compute the error, use the error to adjust the parameter values, then we feed in more training data. That's kind of how this, the amazing thing is this stochastic gradient descent works, even when you are using a batch size of a few images at a time. So even though I'm giving this model 2 DNA sequences and 2 maps at a time, it can reliably enough say, how should I update my gradients or update my parameters to make better predictions at the next time point and converge to something that actually does nicely for predicting Hi-C maps. If anyone's really interested in this, this is a really nice graphical insight, this distill pub on momentum for understanding how stochastic gradient descent works, and these different parameters can influence how your training updates and how far you are from the true loss. The true data can be best used to kind of move your model towards a local optimum. Check it out; it's very cool. So another thing that's important in these machine learning models is instead of just optimizing the objective function, this mean squared error, which I've written right here, right? Instead of just optimizing the true map minus this function of DNA that we've learned squared, we often include some penalty terms that help stabilize learning in the model. So usually stabilize things like the squared weight of all the parameters in the neural network. The other common trick that we use to train neural networks is this dropout layer. People use it in computer vision, natural language, also use it in genomics. It just randomly sets some set of units to 0 with a frequency rate at each step during training time, just reading off the Keras documentation for this layer. So they use it, we use it. It helps models avoid overfitting and reach better solutions. The third thing is actually kind of one of the most incredible to me aspects of how all this works is this idea of early stopping. So we often talk about splitting up our data into training data and test data, but actually what we do in practice is we split our data into training data and test data, and then in the training data we actually hold out some of that data as a validation set. Now, we're not allowed to peek at the test set, right? You can't peek at your test set if you want a fair evaluation of your model's performance, but you are allowed to peek at the validation set and use that to decide different sorts of things. Specifically, you can look at the validation set every time you run through do a full pass through your data set. And when that stops getting better, you stop the training. And this empirically is very good on avoiding overfitting in models and making models that generalize well to the test set and to new tasks. So it's a really simple concept, but also kind of very interesting theoretically. There's a lot of work on trying to understand why this is so useful. Okay, one thing about debugging training. So here is how your performance as a function of passes through your data set might look if things were going well. Epoch is just a jargon for one pass through your training set. You would see your training loss plummet. You're happy. You also see your validation loss plummet. There's some noise, but it sort of eventually asymptotes, and it hasn't stopped getting better by a certain amount of time. You've performed early stopping. You're done. You take the model kind of at the early stopping time point, and that's your trained model. What if your training loss doesn't look that good? So the first time you train a neural network on some new data, you might get something like this. All sorts of things could be going wrong. Something's going wrong. So this is the first hint that something's going wrong. So you can, if your training loss got better, okay, but then it just plateaued at some value that's maybe not very good compared to what you were hoping for, and you can see your validation loss never really decreased. So that means your model just learned junk. So that's kind of a common part of neural network training is neural network loss function debugging. Okay, so with that, I'd like to return you to your groups and have you start in the notebooks on the training section of the collab. How many groups got through the training section already? If you got through the training section already, you can kind of write questions to me. But we have until 5. So let's take 25 minutes, and then I'll collate questions and answer them in the last 10 minutes, and then that will be it. All right. Okay, last Q&A session. We have about 6 minutes left. So I will go over a few questions that we got, and feel free to raise your hand and ask another if you have a burning question. I'm happy to try to answer it. Okay, so what were these individual coolers or kind of whole data sets? So the data that I listed were individual coolers from a few different experiments. So those were 5 individual coolers processed as in the way that Sasha had described at the start of the workshop. Second question, is this a solid amount of data for kind of a neural network model? And that's a good question. So I think in terms of the DNA sequence, and how could you get more data, right? So in terms of the DNA sequence, it's fairly similar among different people. So it may or may not be that useful to collect different IC data sets from different people in the interest of training different models. One thing that we've looked into a little bit, it helps a little bit, is actually taking data sets from different species. So human and mice, and maybe there'll be other species that have good data sets in the near future, and those could be added, and we could learn a richer sequence representation. Of course, then you have the challenge of how do you deal with things that are different between species, and one thing that seems to be different between species is the number of retrotransposable elements that have invaded the genomes of different species. So those could be more challenging to understand with this sort of neural network approach. Another question, came up was how do you design the training test sets? So we split our data into 8 portions, and we used one portion for the test set, one portion for the validation set, and the rest for the training data. And then one cool thing that you can do with that is you can actually train an ensemble of 8 different models where each one is holding out a different test set. And then if you have a prediction you want to make, you can average your prediction over those different models that have each seen slightly different types of data and get a slightly more robust prediction. Another question was momentum and learning rate sound kind of similar, different parameters of this optimizer. Answer is yes, they're kind of similar in a sense. Learning rate is how much each step should you update. Momentum is kind of how much should you care about the last few steps. So yes, kind of they look similar, they are kind of similar. You can change both of them and optimize both of them as hyperparameters on your validation data set, and that can improve model performance a little bit, at least only a little bit in our hands. Okay, and then how to choose sort of a right, optimal, or sufficient number of parameters. There's no general theory on that as far as I know. So I can just tell you what we did, which was we took parameters from literature that worked for other genomics problems. Then we said, okay, well, we have a more memory intensive prediction task. So let's shrink our network a little bit and then try to make prediction. And actually, we first built models for like 250 KB, then half a megabase, and each time we shrunk the size of the network a little bit more, and eventually got something that could work with a megabase. Any burning questions that I didn't address for the second half of the tutorial, or even the first half? I'm happy to take them. If not, I'm happy to take questions offline. Yao and Paulina are also here all week, so looking forward to interacting with you. Thank you. Thanks to Yao and Paulina as well. They have posters you can check out. This is our department from Griffith Park in LA. It's actually pretty nice for biking. Believe it or not, LA has some nice biking. Come ride a bike there. And thank folks in Open2C, because some of the data pre-processing that we did really does depend upon those tools, as well as our collaborators, David Kelly at Calico, and my postdoc advisor, Katie Pollard, was at UCSF. Thanks again.